Here we are going to see the lab diagnosis of different fungal infections. This topic is covered under the following headings. That is the specimen collection, microscopy, culture, culture identification, immunological methods and other methods of identification. So for the lab diagnosis, we have to collect the sample. Depending on the site of infection, skin scrapings, hair, nail, tissue biopsy material, cerebrospinal fluid, blood, pus and body exudates. These are the different samples which we are collecting. Now the direct microscopic examination, in which the wet preparation microscopy is done, under which the KOH wet mount microscopy is done, in which the KOH is used, that is to dissolve the fungal element. And other than that, for the nails 20 to 40% KOH is used because it is the harder material in comparison to skin or hair where the 10 to 20% KOH is used. Then DMSO is used, 10% glycerol is used to prevent the slide drying. After the KOH wet mount, there is lactophenol cotton blue wet mount microscopy, in which again lacto is a lactic acid preservative for the morphology of fungus will be intact. Then phenol is as a disinfectant, cotton blue will give the color to the fungal element. Other than that, wet preparation we will be using India ink or nigrosin, that is the negative stain, and this is used for the demonstration of encapsulated yeast. Next, after the wet mount preparation, we have the smear preparation. So in the smear preparation, the gram staining is done, and the gram staining is to stain the yeast and the yeast-like cells like the Cryptococcus and the Candida. Then the biopsy examination, in which hematoxylin and eosin stain will be used, and this is the most common stain. Here we can see the demonstration, like the fungus will show the pink cytoplasm and blue nuclei. Then the periodic acid shift stain, that is also the fungus will give the deep blue color and the nucleus will be blue stain. Then Grocott Gomori methanamine stain, that is the GMS stain, that is also for the biopsy material. Here it will stain both live and dead fungus because it is going to stain the polysaccharide component of the cell wall. So this demonstration of this GMS stain is also here. Next is the mucicarmine, that is also one of the important stain, that is for the carminophilic cell wall fungus. So here we can see the stain cell wall will be the dark pink color. Okay, here we can see. Now last stain in this category is the Mozon pentana stain, that is the biopsy material it will be used, and this is for the melanin producing fungus or the pigmented phaeoid fungi. So here we can see the brown fungus will be there, or it could be brown or black in the color. These are the different examples in which this stain is used. After staining, there is another method for the diagnosis, that is the culture. So culture media, that will be depending on there are certain growth characteristics of fungus, as the fungus is a eukaryotic cell, so it will take more time to grow. It is a slow growing organism, slow growing individual. So what happening, why the slow growing? Because it's a complexity in the cell structure. Okay, so these are the multicellular structure, more complex morphology is there. So because of that, slow reproduction rate is there. So it will take more time. So incubation time for the growth of the fungus will be more. Now why these are the different significance, why we are going to do the culturing. Now the conditions for the fungal growth. So temperature, oxygen, mostly they are aerobic, pH, and for maintenance of the low temperature, BOD incubators are used, and antibiotics are also used, that is to prevent the contamination. So after the characteristics of the culture, growth characteristics, we will go to the fungal culture media. So let's see, the routine culture media which is used is the SDA, that is Abroad's dextrose agar. The composition is the dextrose, peptone and agar, and these are the specific pH. So here we can see every significance of all the components of the media. So here we can see these are the plates, like and it is a selective culture media that promote the fungal growth while inhibiting the most bacteria due to the low pH. The neutral Savarot's dextrose agar is also used, in which the pH is 7.2 and the peptone will be neopeptone here, and 2% dextrose will be used. Other media are the nutritional deficient media, that is the cornmeal or the rice starch agar media. These are mainly for unfavorable condition, like for the stimulation of chlamydospore production. that is for the identification of a species of Candida. Okay, so after the nutritional deficient media, there is a chrome agar, that is a chromogenic media. So as the name indicates, it is a selective and differential media, and this is for the speciation of Candida. So different species of the Candida we can grow in this media, and they will by their selective color, and that we can differentiate the different species of the Candida. Here we can see, so Candida albicans, Crozia tropicalis, glabrata, and Dubliniensis. Here you can see these are the different color, and this is the culture plate. So after the chrome agar, there is blood agar also for the pathogenic fungus. Then brain heart infusion agar is also enriched media for fastidious or the pathogenic fungus. These are the examples. Then Niger seed agar it is also, that is for the Cryptococcus fungus. So these are the different cultures. Now after the culture, when we are going it on the culture media, how the growth will obtain. So by observation of the growth, we can see the macroscopic or the gross appearance of the fungus will be obtained. So that is also on the rate of growth. Depending on the rate of growth, the growth could be rapid or slow. So if it is rapid, that means if it is taking less than 5 days to grow, so that is called as a rapid, and slow will be taking 1 to 4 weeks. These are the examples. Then the pigmentation could be, we can observe the pigmentation from the front or back. Then the texture could be yeast-like or glabrous. Then velvety growth will be there. Cottony structure will be there. Granular or powdery growth will be there. So, and after that, the colony topography. The topography could be flat like this. Then rugose, in which the grooves are there. Then folded. Then crateriform. Then variocose and cerebriform. So these are the different topographical characteristics of the colony of the fungus. Now coming to the microscopic examination of the colony. Again we will see the lactophenol cotton blue teased mound, where we can see these are the fungus, and nature of the hypha will be septate or aseptate. So these are the characteristics which we are observing here. Color could be hyaline or phaeoid, size could be narrow or wide, types of sporulation could be present or not. Then after that, LPCB, lactophenol cotton blue cellophane tape mound. So in this what we are doing, we are using the cellophane and keeping it on the culture, and then we will put it here, and after that we will get the morphology. Now the slide culture is also one of the important methods. It is the most difficult method, and what we are doing here, we will put the slide, and like this we are putting the bent glass rod in the petri dish. Then we will put the slide on it. Then we will put 1 square centimeter of this media square, and then we will put it on the slide. After that we will put this cover slip, then we will inoculate the 4 fungal strains on the 4 corners of the slide of the agar block, and then we will block the cover with the cover slip. Then we will cover it with the cover slip and incubate for 25 degrees for 48 hours. Then we will put the 1 drop of LCB on the slide and transfer the cover slip to the slide. Then we can observe it under the microscope. So after the transforming this cover slip under the slide, under the microscope, we can see the colony. Now other methods of the identification will be these are the specific method for the specific organism, like for the Candida, germ tube test, Delmau plate test, carbohydrate fermentation and assimilation test. For dermatophytes, hair perforation test is there, dermatophyte test media and dermatophyte identification media. Then Cryptococcus, for the Cryptococcus we have urease test. After these culture, microscopic observation, specific tests, there are certain immunological methods. So in these are the antibody detection. So under antibody detection, these are the tests. Then antigen detection. So these are the different tests for the specific to the different fungus. Then immunohistochemistry, then molecular methods, fungal metabolite detection like by the gas liquid chromatography, in which we are detecting the metabolite. Then delayed hypersensitivity test, that is the skin test. So this way we have seen the complete different methods of the lab diagnosis, where we have covered how the fungus can be identified in different cases. Okay, thank you so much.